Okay, so good morning and good evening to everyone from around the world joining us for this month's session of microTAStic. Today we have two really exciting speakers, so we'll jump right into it. So first up, we have Professor Anderson Shum, and he earned his master's and PhD in applied physics from Harvard and is currently a professor of mechanical engineering and a core member in the biomedical engineering department at the University of Hong Kong. His research kind of spans emulsions, biomicrofluidics, soft matter, and he's received numerous awards, kind of too many to go through, but some highlights. The IEEE NanoMed New Innovator in 2018, Young Scientists Award at the Microsystems and Nanoengineering Summit, and recently was a Hong Kong Engineering Science and Technology awardee from the Hong Kong Academy of Engineering Sciences. So we're thrilled to have him today and telling us about nature-inspired microfluidics. Take it away, Anderson.
Thank you, Julia, and thank you, Cindy and Angela, for the kind invitation as well. This is a great honor to be able to speak at this wonderful seminar series that's within our community that's very close to our heart, microTAS and microfluidics and microchemistry, etc. So I'm from the Department of Mechanical Engineering and also the Advanced Biomedical Instrumentation Center at the University of Hong Kong. Today I'll talk about some of our works on microfluidics that are a bit different from the type of microfluidics that I have been doing when I was a student, and we are trying to find some new inspirations from nature that I'd like to share with you. So as mentioned by Julia, I did my PhD with Dave Waits in applied physics at Harvard. So there we do a lot of microfluidics. We mainly made droplets. We use glass capillaries. We use PDMS to make microfluidics, to make droplets, and use droplets for all kinds of things, materials fabrication, biotech, etc. And so recently, I think just yesterday or these 2 days, a review paper that was written by Dave and I was just published on Lab on a Chip. So if you're interested in some of the things that we have done and some of our views on the developments of the field of microfluidics, please go there and read the article, and we'll be able to share with you some of our thoughts and look forward to hearing from you as well. So these are some of the microfluidic tools that we have been using. I think for this community, we don't have to introduce microfluidics. Essentially, it's just a way for us to manipulate fluid flow through directing them in very tiny channels. So in this talk, I'll talk about some of our inspirations from nature, particularly from membranous organelles in biological cells, from the vasculature in our body, and also from plants. So first of all, I'll just dive right into the membranous organelles part. So as many of you know very well, a cell has many organelles. Many of these organelles has membranes, such as the nucleus, mitochondria. But recently, in the past decades or two, it has been discovered that there are lots of also membranous organelles. Essentially, these are droplets, liquid droplets that are without a membrane within the cells that are also playing very important role in different processes, metabolisms, and other processes in cells. So essentially, because these are membranous organelles in a predominantly aqueous environment, you can think of them as liquid droplets. In an aqueous droplets in aqueous environment. So how is that possible? So this brings us to the topic of aqueous phase separation, which was also discovered quite a while ago and used by people in the green chemistry. So one very common system that people use is the aqueous two-phase systems. This is an example of aqueous phase separations, which can be broadly divided into segregative and associative. And in the case of aqueous two-phase system, this is segregated phase separations. If you have water with two coexisting or more polymers, even though both polymers can be soluble in water, but at high enough concentrations, because there are non-specific interactions between these polymers, typically van der Waals or other that are between neutral polymers. So at low concentrations, they will be just forming a single homogeneous phase. But as you increase the concentrations, you can go from a single phase to a two-phase region. And so For example, here with PEG and dextran, so polyethylene glycol and dextran, at high concentrations in water, you'll get two immiscible aqueous phases. One will be rich in PEG, the other one will be rich in dextran. And this also can be done to a lot of other systems as well. For example, between PEG and sodium carbonate as well, you can clearly see an interface between these two immiscible aqueous phases. And microscopically, this is another example. So we have a droplet with PEG and dextran, and water evaporates so that PEG and dextran becomes more and more concentrated. You can see that small droplets start to nucleate and they start to coalesce with each other, forming two phases. One is tagged by a green dye, the other one is tagged by the red dye. You can see after some time you have two clearly invisible phases. So these invisible aqueous liquid interfaces are very different in properties. So some of the properties include, if you imagine, if we go back to this diagram that I just showed, so you can form two phases by evaporating water, by concentrating the PEG and dextran, but that also means that you can remove the interface, you can reduce the interfacial tension by dilution with water. So you can then go from the two-phase region to the single-phase region. And as you enter the single-phase region, you no longer have an interface. Essentially, then you have zero interfacial tension. And when you have zero interfacial tension, that means the interface is no longer very actively trying to reduce its surface area. So there's a very low interfacial tension. And this passive interface would then be able to respond to perturbations in the environment. For example, even something as minute as sound in terms of a perturbation, it can be detected. So the pitch of this the pitch is corresponding to the wavelength, and you can see that the interface would corrugate with that wavelength as you play sound to different pitches. So this is what's happening to a aqueous liquid jet in an aqueous environment. And the same can be true for music. So basically, essentially, the pitch will determine the frequency of the propagation, and the volume will determine the amplitude of the corrugation. This is only possible because the interface is now so passive. It is not actively suppressing any perturbation. So we took advantage of this and worked with some artists some years ago to have a trying to record the buzzing sound of bees along these liquid-liquid interface in aqueous environment as well. So with that, as I mentioned, I was working on droplet microfluidics. So one key part is we have to be able to make monodispersed, uniformly sized droplets. But because interfacial tension is so low, jet is favored. So in order to form droplets, we have to introduce perturbation to break the jet into droplets in more controlled manner. But this is something that we can deal with. So this was still using glass capillary microfluidics. How can we get inspirations from this to make different kind of microfluidics? So that brings us back to the interface. So now this aqueous liquid-liquid interface, you can approach the interface from the two sides of the aqueous solutions. And you can, for example, add positively charged polyelectrolyte from one side, negatively charged polyelectrolyte from the other side. And when they meet at the interface, you can then form membrane. This becomes very convenient because you no longer need to have a solid particles and then coat the polyelectrolyte layer after layer after layer and then dissolve the particle away. You can just use an aqueous droplets as a template and then add your polyelectrolytes from the droplet and from the continuous phase. And when they meet at the interface, they form the first layer. And then you can add multiple layers, as many as you need. And you can see that indeed it gets thicker and thicker. And after a while, if you need to use the capsule, you can simply dilute with water and wash it, and then you can get your capsules very conveniently. This is an approach that you can also apply to different kinds of polyelectrolyte. For example, some that are pH sensitive. So they will change their properties as you change the pH. For example, if you have a polyelectrolyte that favors the other phases, for example, you add polyelectrolyte to the continuous phase that likes to go to the droplet phase, and add the polyelectrolytes that like the continuous phase to the droplet phase, so they would move towards each other and meet at the interface, then you can form the microcapsule. or you can add polyelectrolytes that like the droplet phase. And then after a while, the polyelectrolyte from the continuous phase diffuse into the droplet. Then instead of the capsules, you form a microgel particle. So these are all possible. And so one of the things that we were doing by my former PhD student, Shipe, in collaborations with Professor Tom Russell from UMass Amherst and Lawrence Berkeley National Lab, is to use these interfaces to assemble membranes. In this particular case, we form membranes that are assembled by combining the polyelectrolytes and nanoparticles. And in doing so, we can, for example, form colloidosomes at these liquid-liquid interface. And these colloidosomes, they are made up of magnetic nanoparticles and graphene particles and glued between by the PAH, a polyelectrolyte. And so at the interface of these liquid protrusions, we have a lot of these magnetic responsive colloidosome. So because the membrane is very, very thin and it consists of materials that can be magnetically actuable. So you can then form a structure like these that are very, very soft, very deformable. You can change the shape as you want by magnetic field. You can, for example, use it to grip an object, move it from one side to the other, and then later on release this. So all of these are very much doable with this. And it is important to remember that in for these structures that we are changing the shape, they are hollow inside. So you can still use their inside to store and encapsulate active ingredients that you like. So in the next example, we were encapsulating some photocatalyst and let these little tubes to go through this maze to the area where we have the red dye. And then we can release the photocatalyst there to cause decolorization. So it is a way for us to come up with new little device that are printable, has very thin membrane that can be magnetically actuable, and because they are highly deformable, so we can get them to go through tortuous path to target location. So this is one of the applications that we called aquabots or aqueous robotic-like structure. And another structures that I'm going to demonstrate is inspired by the vasculature. So this is done by my PhD student Ya Feng and has just been published recently. So conventionally, we have been using microfluidic devices like this one. So on the left, we have a PMMA polymethyl methacrylate device that 3D printed. On the right-hand side, we have a PDMS polydimethylsiloxane device that we can connect. So when we meet students, when we give examples of microfluidic devices to students, we often talk about, okay, actually in nature, even in our body, we have a lot of blood capillaries. We can also think of those as microfluidic channels and microfluidic devices. However, they are qualitatively different from these microfluidic devices that you are seeing from this slide in many ways. For example, the raw materials of these microfluidic devices, the structural materials, they tend to be quite inert and they tend to be quite thick. And once you have fabricated these channels and flowing fluids inside them, it is very difficult to change the fluidic content inside the channels because they tend not to be very permeable. You have to first design your device in the right way, and you cannot easily exchange fluids with the channels, functionalize it, etc. This is very different from what we are seeing in our blood vessels and blood capillaries. You can regulate the flow inside these blood capillaries and blood vessels. For example, diffusing glucose, carbon dioxide, oxygens in and out of these channels. Those can be easily done. So how can we get our channels to be closer to these that we observe in our body, in the vasculature, and in the vascular network? So we take advantage of our all aqueous approach with the aqueous interface as well. So now again, we can do printing. We can do 2D printing, 3D printing into the device, the channel geometry and the architecture that we want. Here we use two components, one the APAM, the other one is a chitosan. We use chitosan as an ink and print this chitosan's aqueous solutions into APAM solution. And the chitosan and the APAM will meet at the aqueous liquid-liquid interface and form a membrane. So now, depending on what structures you've printed, you can then get these components to form a very thin membrane wall. So now the wall of the microfluidic device that you print is no longer like the PMMA or PDMS that are very thick and very inert. They can be very thin and very functionalizable. So for example, this is the shape that we have printed. You can indeed see that the red dye can flow from left to right. The inside we have the aqueous solution. Outside we also have aqueous solution separated only by a very thin wall. And with that, we can also see that you can do all kinds of different things. So now, for example, between the left side and the right side, you can flow to two different solutions with different color. And you can also, for example, later on, direct some of the fluid from outside this device. For example, let me see. So if we play this video, so if I add a droplet on the outside, some of the dye can be directed outwards, diffusing across the membrane interface to this newly added droplet. And you can at the same time also, so you can see the yellow dye and a little bit of the red dye going to this originally transparent dye. You can also add another dye, for example, blue dye from to here, and the blue dye would diffuse into the channel, and you can see a little bit of blue and green already. So this allows exchange of fluids after fabricating the device. And then another thing that we can do is instead of just adding dyes, we can add other functional components such as enzymes. So with these enzymes, for example, we can add it to 4 parts that are tagged here. And then you can see in the next video that we flew, so these enzymes can decolorize the green dye, but not the red dye. So you can see that as the red dye and the green dye flow through this channel, the green dye passing through this region with the enzyme that was functionalizing the channel, there's no longer green color after passing through it. And the other parts can go through those. So this provides a very convenient way to first make a microfluidic channel and later on exchange fluids between the contents inside and outside, and also functionalizing different parts of this channel as you wish at different time and at different point location of this channel. So this was inspired by the vasculature. So the last inspiration that I like to talk about was by plant. So this is a work that was done by my former PhD student, Pan Yi, who is now having his own independent group in the Southwest Jiao Tong University in Sichuan. We call this the TOM, the TOM device, because it can be transformable. It has the shape of an origami, and we have microfluidic flow inside, so we call it trans-origami microfluidics. So if we look at clover leaves, during daytime, it can open very widely. And during nighttime, it can close very widely. This is very efficient configurations because for photosynthesis and also for other photochemical reaction, when the device or when the leaf is completely open, it can capture the largest amount of light. But then when there's not so much light, you can close it to minimize the energy consumption by respirations and other processes. So when we think about our microfluidic device, typically once we've made them, we cannot change their shape. We cannot change them like these leaves. So in order to make that possible, we have to, for example, first make materials that can respond to the environment. So Pan Yi was trying to combine two types of materials. One is hydrogel, the other one is elastomer, and functionalize the hydrogel and the elastomer. So by doing so, we can put in different components that respond to the environment, respond to light, respond to humidity, etc. So these are some examples. For example, you have a hydrogel and elastomer, and because the two material responds to the environmental stimuli with different degree of change in the volume. So then you will cause bending of the materials. And if you use these materials to form arms or petals in a structure, you can have different kind of motions, directional motion, non-directional motion. The different arms can move in a synchronized manner or non-synchronized manner. So here are some examples. If we have a petal, like a flower shape, that responds to light, we can cause this flower to open and close in response to light. And we can also, if we decide a different shape and apply to different environment, we can also use it to grip objects from one end to another end. These are synchronized motion. All of the petals are moving simultaneously in a synchronized manner. And you can also have a synchronized motion. For example, you shine light in different location, and you can get this structure, this shape to move forward. And also, we can, besides light, we can add other stimuli. For example, a hydrogel that will respond to the humidity. So when you do that, here, this is a caterpillar. You can change its motion by shining light, adding water, shining light, adding water, and cause it to move because the two arms are moving in a non-synchronized manner. So they are inching forward and backward. So this another way. You can also have more non-directional movement, such as this jumping, or you can have this little object to jump out of an obstacle, which is a ring in this case. You can cause it to jump, triggered by the environment. So Panyi was using these kind of concepts, but add a layer in between with channels to do microfluidics. So this is an example of the design. If we put in the functionalized patches in different locations, in this case, we follow the shape of origami so that they can fold and unfold in a more organized manner, and build in fluidic channels in between. Then you can flow fluids into these channels, and then the device that you've made in the end can open and close. We can think about stimuli such as temperature, humidity, light. So here is an example of a microfluidic device built this way. We have the channel. You can see the fluid flowing in with light and also low humidity. This device can open. And if we later on reduce the light and also increase the humidity, it will close, like you can see here. So trying to think about what we can do with this type of device, we again get inspirations from the plants to try and do a photochemical reaction. So when there's a lot of light, we open the device, and when there's not so much light, we close the device. So this is the concept, and we try to make a device that has a self kind of built-in feedback loop. When there's light, it will open. When there's no light, it will close. And we try to compare this TOM, transfer-origami microfluidic device, with a device that's always open and that's always closed, and compare the reaction conversion and the overall consumption of energy. So you can see that we just do this outside our lab, in our building, in open areas, as well as in the lab. You can see that when they're depending on the amount of light in the environment, they would open or close. And if you compare the conversion of the reaction in the end with a freely kind of transfer open and closing device, we can have higher conversion than the device with a fixed shape, with the same channel shape. Obviously, if you have a device that's open, you have a higher conversion than a device that's always closed. But we demonstrate that if you make a microfluidic device that can respond to the environment with such a feedback control, you can maybe have more effective photochemical reactions. So we are still trying to think about how to take advantage of this for other applications as well. And with that, I'd like to conclude that we have been inspired, and we have been doing a lot of microfluidics. It's a very exciting field. And just moving forward, we look at our environment, look at the nature, membranous organelles suggest that we can also think about assembling devices using aqueous liquid-liquid interface. Vasculature inspire us to think about devices that you can have exchange of fluid between the inside the channel and outside. as well as making device that can be functionalized on demand. And plants like cloverleaf inspire us to make devices that are responsive to the environment. So with that, I'd like to thank you all for your attention and also thank all the funding agencies. And obviously, I have done none of the works. All of the works are done by my current and former students. And so I'd like to thank them as well. So thank you.
Fantastic. Thank you. So we have a number of questions in the chat and limited time for them. So I think maybe what I'll do is I'll read them out and then hopefully we'll get to all of them. So
Sure.
the first one is in relation to the vasculature and membrane-based microfluidics work. What's the smallest diameter and inner dimensions of the channel that you can fabricate that way?
Yeah, so at the moment, this is mainly limited by the printing. So if we can print it, then we can typically form whatever shape we want. And obviously, we have to stop the assembly, because if we leave the material there for too long, it will get thicker and thicker. But that is also quite easy because we can just dilute it away. So it's a optimization of the assembly time between, and also concentrations of the components that we add to the ink and the surrounding environment. And then besides that, it's just about the dimension of the nozzle you can use and how much you can print.
Got it. Okay. The next question, I should have said the first one was from Sujit. The next one is
Thank
from
you.
Seth. How much pressure can the membrane withstand?
That's a great question. Indeed, with the very thin membrane that we're using, it cannot withstand too much. There are probably ways that we can reinforce the channel wall, which we have. I mean, for the proof of concept, we have not demonstrated that in the panel, but what you can do is you can first fabricate a membrane and test its mechanical properties off chip, and then you can use a similar kind of strategies and formulas to fabricate a device and a membrane that you need.
Got it. Next question is from Myra. Is there a limit to the types of fluids you can flow in the membrane fluidics? So are there any fluids that might disrupt the membrane that forms the channel walls?
That's a great point. So currently, with most of the fluid that we flow into these membrane channels, we are using non-interacting fluid with the channel wall. Because as I demonstrated to you, we can functionalize the wall. That means if we flow in solutions that will also interact with the wall, we can also change the properties of the wall. So that sometimes can be an advantage, but if you're just trying to use the channel geometries that, for flow purposes, then that can be disadvantage because the channel properties can change over time. So but the advantage is that there are so many combinations of aqueous soluble compounds that you can use to make the membrane. So depending on what properties you want, you can always tune it. You can first test it off chip before you do the printing, and then you can just print it afterwards.
That makes sense. Next question is from Bin. Does the channel have to be used in solution? And will the fluid dry out if exposed in air for a long time?
Great point. So with our current works that we have done, indeed, if they dry up, the channels would not be functional. So we had to at least keep them humid and keep them with moisture. But they don't have to be completely soaked. If they are moisturized enough, that's already okay. If we want to use the such devices in a dry environment, I imagine we can use the printed channel with the wet membrane first as a template, and then we can coat with other materials, and then probably we can use it dry, but we have not demonstrated that yet.
And then the last question here so far is from Kakit. Sorry if I'm destroying your name.
Thank you.
The question is, are the printed membrane structures suitable as cell culture scaffolding?
That's a good question indeed. That is something that we are trying. We believe that we believe that using that as a scaffold, we can potentially build larger kind of cell cultures or even tissue or organoid type, and we can direct fluid in and out on the surface. So if we believe that if we functionalize the surface of these
Yeah, I just put
membranes,
it on. Yeah, no,
that is
no, but
what
this
we're
is
yes. So that is something that we're working on, and we have really high hope, and we believe that this will work.
It's awesome. So we know that you have another engagement to go to. So I think we'll wrap up questions there. Let's thank Anderson again for a great talk.
And thank you, Julian and Julia, and thank you, Cindy and Angela and all the organizers. It's a real pleasure. So please feel free to contact me if you have more questions, and I'll be very happy to meet all of you in person as well.
All right, awesome. So next up, we have our second speaker of this double header today. So Tae-Eun, if you want to go ahead and share screen. Um, Tae-Eun Park is an associate professor at Ulsan National Institute of Science and Technology in Korea. She earned her PhD in 2015 from Seoul National University, and she developed brain drug delivery systems there from 2015 to 2017. And during her postdoc at the Wyss Institute, she started working on these microphysiological systems that you'll hear about today. And now, as faculty, her research is concentrated on these humanized organ mimetics using microphysiological systems and organoid technologies, and also investigating novel therapeutics. Go ahead, take it away, Tae-Eun.
Yeah, thank you for a nice introduction. Very good to see you all. I'm Tae-Eun, and from Department of Biomedical Engineering at UNIST. And today I want to talk about the microphysiological system technology for the enhancement of the drug delivery system. So let me quickly. Sorry for the technical problem. Yeah. So FDA Modernization Act 2.0 promoted some transition toward the non-animal testing method for the regulatory using the drug development, and then alternative methods such as a microphysiological system and an organoid like offer the improved efficacy and then reduce the cost for the preclinical drug testing. So the primary focus area for advancing the MPS technology, I would say, in the future could include some issues. Is the following, like the first issue is development of the integrated multi-organ MPS to enable the study of the interaction between the different organs. And then second issue is enhancing the data collection through the incorporation of the biosensor and then some actuator within the MPS for comprehensive monitoring. And then third issue is implement of the rigorous quality control measure for the MPS through the analysis of the in vitro and the in vivo correlation. And the fourth will be establishing the uniform standard across the MPS system for consistent drug testing. However, when I delve into the potential of the MPS technology, I find myself asking a critical question, like how effectively can we use the human MPS not only for testing drug, but also to discover the new target for the drug therapy and to innovate the drug delivery mechanism and the strategy. And then today's talk is about the use of the human MPS to discover and then enhancing the drug delivery strategy. I would say the greatest obstacle in the field of drug delivery is the administration of the brain drug to the human brain, which involved in the overcoming the very protective physiological barrier, which is called the blood-brain barrier. I'll call it BBB from now on. The BBB is a very selective and semi-permeable and then protective barrier, protective border that separates the circulating blood from the brain in the central nervous system. So because of this, most of the drug cannot enter the brain. This strong physiological barrier limits the penetration of most of the brain drug. And ironically, as our understanding of the disease mechanism deepen, the size of this drug tend to increase. So it's more difficult to make the drug get into the brain. So one of the most promising strategy to overcome this problem is using the Trojan horse strategy. The Trojan horse strategy is like making drug encapsulated or decorated with some ligand, and then making this ligand exploit the native brain molecular transport system, and then making drug together get into the brain. That is the Trojan horse strategy. For this, the receptor-mediated transcytosis called the RMT has been explored as a method to transport the molecule and the drug across the BBB using the brain shuttle, like a monoclonal antibody targeting the transferrin receptors expressed on the brain endothelial cell, which are less restricted by the size and the shape of the carrier. So no matter what, like it can be like a really big, the drug, it can be like a 500 nanometer. The size of drug can use this RMT to cross over the BBB. But despite the promising animal model result of this BBB shuttle or BBB ligand, most BBB shuttles fail in the clinical trial, and often due to their size and complexity and then expenses. And then I will say this may due to the lack of advanced and then more realistic and humanized in vitro BBB system in their development. So when I was in the business student at Harvard University, as my postdoc, I was focusing on developing the human BBB MPS using this simple microfluidic device. This device has a two microfluidic channel. The bottom channel is microvascular channel, and then upper channel here is the brain channel. The bottom vascular channel is covered by the iPSC-derived brain endothelial cell, which is a very open channel. And the other side, the brain channel, you can find the human, the brain, pericyte and astrocyte. Those are critically important cells to support the function of the brain endothelial cells, the barrier function. And then to enhance the BBB functionality of this human BBB MPS, we used two strategies, the stem cell engineering and the microfluidic approach. The stem cell engineering means when we have developed a methodology for the differentiation of the brain endothelial cell from the iPSC by controlling the oxygen condition, which is inspired by the developmental the microenvironment. And then also we have used the physiological shear stress. We offer the physiological shear stress to the brain endothelial cell to enhance the functionality. So by using this two approach, we could obtain the in vivo BBB-like physical barrier function, which means that barrier integrity of the brain endothelial cell is very similar to the physiological the BBB function. So it's not leaky at all. While the control cell is just iPSC-derived brain endothelial cell without oxygen condition, they lose their function within 24 hours. But in our system, we could maintain the physiological barrier function for 7 days. And then another important part is discovered is shear stress is extremely important to have a metabolic barrier function from the efflux pump expressed on the brain endothelial cell. We found that shear stress is critical for the activity of the receptor and then transporter and then efflux pumps. And another important part is because we have a very tight junction brain endothelial cell, and then they have well expressed brain-specific receptor and transporter under shear stress. We could recapitulate the shuttling of the BBB targeting antibody, and then they created a nanoparticle. So I was thinking a promising approach involving using a human BBB MPS is to screen the human BBB shuttle. And for this, we started to screen the aptamer. So aptamer, also known as a chemical antibody, are kind of favored for their practicality over the traditional antibody or peptide as a BBB shuttle. And then traditional selection method to screen the aptamer, they used to identify the BBB shuttle aptamer, like those targeting the transfer receptor, often select the aptamer. They fail to target the protein in their native cellular state because they just immobilize those receptor on the solid state. An in vivo evolution strategy in mice has tried to address this, but difference between the mouse and the human BBB complicated human application of those BBB shuttle. So therefore, our human BBB MPS based selects can be the promising for selecting the aptamer that are effective under human physiological condition. So we have developed a human MPS selection method for identification of the BBB shuttle aptamer. So MPS selects our, what we suggested, consists of the three major steps. First, dosing the single-stranded DNA library to the vasculature channel under fluid flow. And then collecting the BBB penetrating the single-stranded DNA from the brain channel effluent, and amplification of the single-stranded DNA pool for the next round by PCR, like this. And then we repeated this screening for 5 times. And the ratio of the output-input aptamer across the BBB in the first batch of the MPS selects was increased by round of the selects. This implies that the selection of the aptamer has been successful in the MPS. And then we repeated one more time as a second batch. And then this also showed a successful the screening using the MPS. The sequence of the aptamer pool revealed the 6 most enriched aptamer sequences here. And then interesting part is that the top sequences were almost identical between 2 different batches of the MPS selects, right? So when you see the ranking of the BS1, in the first batch was the most enriched one, and the second batch was the second most enriched one. But this implies that the MPS selects using the human BBB MPS provides a reliable and a repeatable screening methodology. And when we analyze the structure of the aptamer among this like 6 top enriched aptamer, the 4 had a stem loop the structure. The next, we verify the BBB penetration efficacy of this top 6 aptamer using the Transwell and then also BBB MPS system. And then we found that the BS1 is the most promising in terms of the capacity to cross over the BBB. And interestingly, even though we used only the BBB MPS, the BS1 showed a BBB specific shuttling capacity, which means that it didn't show the transport enhancement in the liver endothelial cell, lung endothelial cell, or umbilical endothelial cell. We next verify the transport capability of the BS1 aptamer using the streptavidin, the 60 kilodalton protein as a model, the protein drug. And we found that when we decorate this streptoavidin using our aptamer shuttle, it showed enhancement of the uptake in the brain endothelial cell. And then interestingly, also in the astrocyte and the neuronal cell. And this means that the BS1 aptamer we identified, may target the receptor that are common to the human BBB and astrocyte and the neuronal cell. Given that the many receptors on the brain endothelial cell are also present on the brain neuron and astrocyte. And then we explore the mechanism of the BBB targeting and then BBB transport. We couldn't find the the receptor, the bounded to the receptor, what the BS1 aptamer is targeting. But what we could do is we could see the endocytosis mechanism. There are many different endocytosis mechanisms for the BBB, transcytosis mediated molecular transport. And most of the BBB shuttles are using the clethlin mediated transcytosis. So by using the inhibitor drug, by blocking the clethlin and then cabula-mediated endocytosis and the more microphysiological-mediated endocytosis, we found that when we block the clethlin-mediated endocytosis, the BS1 aptamer showed a highly decreased BBB transport the capacity. This means that BS1 is depending on the clethlin-mediated endocytosis. So even though we discovered the HBS1 aptamer using the human BBB MPS, we verified their activity using the in vivo model. When we intravenously injected those aptamer to the mouse, when we collected the brain tissue and then quantified how much aptamer accumulated in the brain, the mouse brain, we could see the high enhancement of the brain accumulation in the BS1 aptamer. And also we have tested if they can to transport the big cargo like a streptoavidin. Compared to the control aptamer with its random sequence aptamers, when we decorate the streptoavidin using the BS1 aptamer, we could see the enhancement of the brain accumulation in the mouse. The microscopic examination of the section, the mouse brain, also revealed the accumulation of the BS1, the molecule at the a BBB and the brain parenchyma. When you see this, the red dot, you can see that the most of the molecules, streptoavidin, is accumulated in the brain parenchyma. It was the same when you see the hippocampus. In the higher magnification, you can see that this green part is lectin, which mark the brain endothelial cell, which is BBB. And then you can see that these red dots are discovered in the brain parenchyma. Okay, this means that they could, the BS1 aptamer allow the influx of the molecule into the brain across the BBB in vitro. It was the same in the cortex, in the higher magnification. And then, as you can see, interestingly, can find that in the control aptamer, most of them are just accumulated in the BBB part, which means that there's no influx of the molecule into the brain. But in the HBS1 aptamer, you could see that they actually cross over the BBB and then they are found in the brain parenchyma. This means that even though we use the human BBB MPS to discover the function of BBB shuttle, it was also working in the in vivo mouse. The next question was, is the BBB MPS the better tool to screen the BBB shuttle compared to the traditional static platform? So we recently found that MPS approach outperforming BBB shuttle screening by replicating the endothelial glycocalyx-mediated molecular transport. What we found is in the under the physiological shear stress using the microfluidic device, the brain endothelial cells show the really beautiful endothelial glycocalyx, while in the static, the BBB transfer condition, there was no glycocalyx decomposition in the brain endothelial cell. The recapitulation of the glycocalyx is very important considering that many viruses exploit this glycocalyx to bind to the receptor firmly and then go through the molecular transport and then enter the host cell. And because in our BBB MPS, it has a well mirrored endothelial glycocalyx, the BBB shuttle molecule we found can it's the same. It can exploit the glycocalyx to cross over the BBB. So this functioned very well in the in vivo BBB because this also have a highly well developed endothelial glycocalyx and the apical side of the brain endothelial cell. So when we screened the BBB shuttle using the MPS, we could see that it showed a really high brain accumulation efficacy by crossing over the BBB, while we discovered the BBB shuttle using the static, the BBB transfer condition, the functionality of the BBB transportation was very low. This kind of approach can be expanded to develop the AI-driven prediction of the functionality of the drug delivery shuttle. And for this, I was thinking we may need a human multi-organ MPS approach because even though we just try the human BBB MPS for the better screening to avoid the liver system or the kidney system and specifically deliver the drug to the specific organ, we need to screen the shuttle molecule using the human multi-organ MPS. And then by developing this methodology, we found that we can get like huge data in terms of the sequence of the aptamer, and it can be expanded to the peptide sequence, antibody sequence, and nanobody structure, liposome, and the cells, and so on. So we could get a lot of data in terms of the if they can cross over the BBB or not. So by using the AI-driven prediction, by just putting some structure of the shuttle molecule, maybe we can predict if they can target and then cross over the vasculature and then to reach to the specific target organ. So I believe that human multi-organ MPS and then drug delivery, the methodology can achieve this like a future. So this is a summary. We have developed a human BBB MPS by the hypoxic stimulation and the shear stress, and especially shear stress was very important for the activation of the efflux transporter and the receptors expressed on the brain endothelial cell. And then based on this success, we could select a human BBB shuttle which can cross over the BBB, which enables the more reliable and repeatable screening of the BBB shuttle. And then this study was conducted with a collaboration with Professor Kim Myung-ju at UNIST and then Tae-Eun Park at UNIST and funded by the government. And thank you for your attention. I'm welcome to any questions.
Yeah, thanks for the great talk. While we wait for a couple questions to come in, I had a question about the attachment of the aptamer to the protein. Do you think that the attachment point at all impacts the shuttling capacity of the aptamer?
Like a specific bind to the receptor is extremely important, and then this is like based on the three-dimensional structure of the aptamer, and then also the specific sequence of the aptamer, and then also chemical interaction between the sequence and then receptor. So it's not easy to just to tell you like one mechanism. It's all combinational.
For sure, definitely.
Yeah.
Yeah, and then I was interested to see the comparison, between the microphysiological model and also, doing the same experiments in vivo. Do you think kind of with the FDA kind of changes to the policies in terms of being able to use these types of systems, we'll see less and less of the use of models that aren't like fully relevant or fully recapitulating particular disease states, or do you find that there's still kind of quite a bit to go and that, reviewers still expect to see the in vivo data, or, what's your perspective on that?
So I think MPS is a quite a premature area, premature field. And then I was thinking there was no like a deep study into the in vitro correlation. And then before this, I guess that it's gonna be quite difficult to, like um applying for this a document without the in vivo experiment. I mean, still, I guess we need an in vivo experiment, but we're still ongoing. Like for example, FDA and then our regulatory like bureaucracy is pushing our scientists to standardize this the MPS protocol, and then I guess like in 5 or 10 years, probably without animal testing, we can, test the drug. And then also, as I suggested, it can be used for the discovery of the new drug target and the drug delivery system.
Yeah, definitely. I think it's super exciting, especially for diseases with poor animal models.
Yeah.
And then of course we have plenty of evidence of uh, diseases with good animal models, just basically helping us produce therapeutics that seem to cure diseases only in the animal model, but
Yeah.
not in the human. So um, are there other questions you can enter into the chat, or also just unmute and ask your question? Group seemed to get a little shy. Um,
I have a question. What do you think is I guess, the biggest challenge in further pushing the MPS system for, like, what do you see like any hesitations in adoption of MPS system, and like what might be the like challenges, and also how to go around such hesitations in using MPS systems, like versus animals, like maybe from like companies or from other uh, I guess, institutions that have been using animals for a long time.
Mm-hmm. Yeah, so it's not easy to go through like a very quick transition between the animal model and the animal methodology. And then actually what I was thinking is still this microfluidic approach is something like challenging for the like researchers in the pharmaceutical company, and then many researchers in the biological decide. And then like one big challenge is the making the MPS for like easy use for all the scientists. It can be very important. And the other side is when you use the animal model, for sure you can see the physiological the response to the drug, and like this like involve the organ interaction, and then how those like molecules like secreted from the one organ can affect the other organ. And as you know, the like a multi-organ MPS is still ongoing, and it's quite difficult because of this practical the problem in terms of the offering the very optimal culture media to the each organ and so on. And then third one is in terms of the PKPD, the MPS system does not fully represent the whole physiological system. And then it's still very challenging to correlate with it, like in vivo, the response to the drug. So yeah, there are many things to, to fix, to make the MPS go to the really like non-animal methodology.
For the multi-organ systems in particular, what do you think is sort of the future in terms of getting the different organ components to self-generate the kind of expected expression of different factors and things that each organ uniquely needs.
Mm. Yeah, it's a very difficult question. If I know the answer, probably I'll go for it. And then if you're just using the like a simple cell line, it's gonna be quite, the easy to make the adapted cells to function. And then ironically, as we understand more like a cells and then physiology, and then the MPS is going to be more complex. It's going to be more difficult to make every cells to be happy in the one linked system. So I don't know. I was thinking like maybe some very well developed biomaterial which are incorporated in the MPS. We like just like generated some the molecule make the cells in the MPS happy in the each organ model will be also very promising. So not only just to rely on the culture media system, we have to also think there's some biomaterial to incorporate in the MPS.
Yeah, definitely. To the extent that we can learn from biology and the impressive macrofluidic system that is our body, there's likely something we can mimic from that can help with that. I also had a question. So going back to the aptamer, so what sorts of cargo are you most interested in being able to transport in terms of interest in drugs that haven't traditionally been able to cross the blood-brain barrier? I think certain like chemotherapeutics definitely that would be of interest. But I'm curious to hear also what you get questions from potential collaborators about, like, oh, can we transport this particular
Yeah, definitely.
molecule
So
or biologic?
everyone's a dream in the field of a brain drug delivery. It's gonna be like delivering the antibody therapeutics right now. Antibody therapeutics is quite big. It's not easy to make it, into the brain. So the researchers are trying to incorporate those BBB shuttle to the antibody, and we're actually working on it. And then another thing is probably the therapeutic cell. It can be more difficult to like transport to the brain. Like many stem cell therapeutics, it's gonna be almost difficult to inject into the like a bloodstream to make it cross over the BBB. But if you understand like how BBB shuttle is working, maybe we can modify those stem cell therapeutics as well.
Yeah. I'm also wondering maybe if I don't know, the cells seem especially challenging, but maybe as an intermediate to the antibodies, like maybe fab fragments are already kind of along the size of what you've been able to transport. Maybe there's some like therapeutic fab fragments that could take a ride on an aptamer into the brain.
That could be that would be nice. Like a small fragment of the antibody can be more useful to be integrated with the aptamer, and then also nanobody therapeutics. Like
Exactly. So we have a question from Yaoping. Yaoping, do you want to unmute yourself and ask your question? If not, I can just ask it. Oh,
okay.
okay. Yeah, she's in another meeting. So yeah, how do you think about the tumor genesis? I guess I don't fully understand the question.
I think she is asking about the risk of iPSC growing tumors, because when you induce the iPSC, that's what I'm guessing from the question.
That makes sense.
Yeah.
Thanks, Angel.
Yeah, good interpretation. Well, so this is not for in vivo integration, and this is just the in vitro system. So I will say there will be no problem of the tumor genesis in the body. And then we have a well-optimized condition for the iPSC differentiation, and then we found that almost no pluripotency in the differentiated brain endothelial cells. So I think we don't need to worry about this issue.
Great. Are there other technical questions for Tae-Eun? If not, also in the meantime, we have a pretty large audience of trainees with us that attend these seminars. And so we're always interested to hear our seminar speakers unsolicited, well, in this case, solicited career advice. How maybe to ask like, how did you kind of come into the research area that you work in now? How did you find what you wanted to work on for your career, and how have there been pivots and in what that direction is? Would be my question to kick it off.
Well, like answering your question. So in my PhD, I was working on the brain drug delivery, and then I was modeling the BBB and so on. And then I found that I always used the transfer system and like a traditional circuitry system. I was wondering, like, what if I have a really advanced the platform to like develop the drug delivery system from the beginning? And then it must be humanized because we always use the mouse model to verify the function and so on. But it cannot be, like be successful in the clinical the trial. So yeah, at the time I was thinking about this, and then fortunately, MPS system, the organ on a chip, has emerged as a promising the field. So I decided to study not only drug delivery, I want to study this organ on a chip system. And then I found that this is really perfect combination. So I was very lucky in that sense. And then when my students ask about like their career plan and so on, I always tell them, don't do like the same thing in your postdoc. Instead, you have to think what uh, what's lacking in your research, and then what gonna be the really like a promising future if you integrate the new technology and a new idea, and then learn something new in the postdoc. And then if they like incorporated those like 2, 3 like very different technology, gonna be very powerful for their the research in their future.
Yeah, that's great. I think yeah, it's always kind of inspiring to work with a tool that is subpar in some way. You see the limitations of it, and you're like, if this could just be better, we could understand so much more, and then that kind of drives you in your path for sure. So I saw one more technical question from June Lee. Would you like to unmute yourself and ask your question? Or are you also in a meeting?
So yeah, so how much be emulated in the MPS similar to that in vivo, like a permeability and so on? Yeah. So when we like measure the physical barrier function using the TR, transendothelial electrical resistance, we found that it's quite similar to in vivo, which means that the barrier integrity of our BBB system, it's similar to the in vivo BBB. And also when it comes to the metabolic barrier function, because there are many efflux pump on the brain endothelial cell, when they when they bind it to the small hydrophobic drug, they pump out those drug from the brain to the vasculature again. And then in terms of the metabolic barrier function, it's not easy to recapitulate in vitro. And then what we found is under the physiological shear stress, the brain endothelial cells have a kind of activated efflux pump, which work for the metabolic like a barrier. And when we tested representative metabolic barrier, it showed a quite a similar result with the in vivo data. So I would say this is, yes, quite the good the methodology to study the BBB.
That's great. I think maybe one more career related question. So there's a lot of trainees that are also enticed towards industry. So I guess I have a two-part question. One is for someone that, hasn't done much MPS work but is really fascinated by it, how should they get started? And then for those that are already interested but are being pulled towards the biotech sphere, what would you say are kind of the advantages and disadvantages to doing research on MPS in academia versus what you see from your colleagues in industry?
I would say that when I was in USA for my postdoc, I could see there a lot of startup company like pushing the direction to the MPS technology. And in Korea, only a few the companies are like doing like a lot of amazing work. I would say that, well, not only like a biotechnology company, also like many big pharmaceutical company like Lonza, and then like many good pharmaceutical companies are kind of employing this MPS technology, and then probably they're gonna need an expert in MPS technology, and also some having some biological the knowledge, and plus maybe if you can do that like a pharmaceutical the side of the knowledge, and they're gonna be also very interesting. In the academia, I would say that MPS field is quite immature, so there are much things we have to do, like not only practically like standardization of the MPS, we have to push a bit more to make this MPS functional. So in that sense, I think academia has to do like a lot of work by collaborating with a company. So if the students are interested in this like MPS field, because the importance of the MPS field is getting bigger and bigger because FDA and many regulatory bureaucracies started to believe the non-animal methodology. So it's going to be definitely promising to like go for the industry. And I would say that it's going to be bigger, and then you just wait for the moment.
Great. Last call for questions here. If there are no other questions, then let's thank Tae-Eun again for a great talk and for sticking around for some Q&A.
Okay, thank you so much. I want to see you all in microTASC this year.